Good cholesterol, bad cholesterol. You've heard these terms your entire life. Your doctor talks about raising your good cholesterol and lowering your bad cholesterol. But here's the thing. It makes about as much sense as talking about good water molecules and bad water molecules. A water molecule is simply H2O. There's no good version of it or bad version of it. It's just water. Well, the same applies to cholesterol. It's just one specific molecule. So in the last video, we exposed how the 1977 dietary guidelines were based on minimal evidence and how a $700 million study actually showed that low-fat diets can increase heart attack risk by 26%. Now, if saturated fat isn't the villain we've been told it is, then what about cholesterol? Well, most people, including many doctors, don't actually understand what cholesterol actually is. They don't understand the difference between cholesterol and lipoproteins. And they definitely don't understand that there's no such thing as good and bad cholesterol. Now, this is cholesterol. This is the actual molecule. And it has a specific structure. It has 4 rings, a hydrocarbon tail, and a hydroxyl group. So when we talk about good cholesterol and bad cholesterol, we're pretty much implying that the molecule itself can be good or bad. But that's just impossible. Cholesterol is just cholesterol and the molecule can't change. Now, before we go further, I need you to understand something that is crucial. Cholesterol, it's not some toxic substance your body's trying to get rid of. It's absolutely essential for life. First thing, cholesterol makes up about 30% of every cell membrane in your body. Every single cell. We're talking your brain, your muscle, your organs. They all need cholesterol in their cell walls to function properly. And then here's something that should really make you think. About 75% of cholesterol in your body, it's actually made by your body. Literally, your liver produces it. So if cholesterol was so dangerous, why would your body manufacture most of it? Well, because cholesterol, it's so important and it's also the building blocks for all of your steroid hormones. So testosterone, estrogen, cortisol, progesterone, your vitamin D, which you make when sunlight hits cholesterol in your skin cells, your bile acids, which you need to digest and absorbs fats from your food. And like I said, it's a critical structural component of every cell membrane. Pretty much without cholesterol, your cell membranes will literally fall apart. You wouldn't make steroid hormones, you would not be able to digest fat, and you would not be able to synthesize vitamin D. So cholesterol, it's not the enemy. It's actually essential. Your body knows this, and this is why it makes most of it internally. But here's where it gets very interesting and where the confusion comes in. So cholesterol is a type of fat, and fat has a problem. It doesn't dissolve in water, and we know that the blood is water-based. So think about oil and water. They don't mix. Well, the same thing happens with fat and blood. If you just dumped free fatty acids in your bloodstream, it would literally clump together. And that's actually a real medical emergency. It's called a fat embolism. Pretty much the fat clumps block your blood vessel and it's very, very dangerous. So your body has this problem. Fat is essential for life. Cholesterol is essential for life. But they can't just float around in your bloodstream. So how do you transport them safely? Well, your body packages fat and cholesterol into special structures that are called lipoproteins. So this is a lipoprotein. And this is what we're actually talking about when we say cholesterol in everyday conversations. So look at the structure. On the inside, you've got the cargo hold. So the cargo includes cholesterol, triglycerides, fat-soluble vitamins like A, D, E, and K, antioxidants, and pretty much all the fat-soluble stuff that need to travel through your blood. Now, let's look at the outside. We've got this complex outer membrane that's water-compatible on the outside so it can travel through your blood, but fat-compatible on the inside so it can hold the cargo. Now, embedded in this outer membrane are proteins, and these proteins act like an identification badge or a signaling device. So keep in mind these proteins because they're going to be the key to understanding cardiovascular disease. Now, there are several different types of lipoproteins, and they all differ based on 3 things: their size, how much cholesterol and triglycerides they're carrying, and what proteins are embedded in the outer membrane. So, for example, we have chylomicrons, VLDL, IDL, LDL, and HDL. but the two everyone talks about are always HDL and LDL. So let's see them. HDL means high density lipoprotein. This is what usually people call the good cholesterol. It's smaller, denser, and it generally transports cholesterol back to the liver. LDL means low density lipoprotein. This is what people call the bad cholesterol, and it's larger, less dense, and it transports cholesterol out to our tissues. But here's what you really need to understand. Both of these are carrying the exact same cholesterol molecule. The cholesterol inside the HDL particle, it's identical to the cholesterol inside the LDL particle. So why do we call one good and the other one bad? Well, it's not about the cholesterol itself. It's about the lipoprotein particle. It's about where it's going, what it's doing, and what happens to it along the way. HDL is generally moving cholesterol back to the liver for processing, for recycling. So we think of it as a cleaning up crew. And that's why it's called good. LDL, on the other hand, is delivering cholesterol to your tissues, which you actually need, is essential for you. But under certain conditions, LDL particles can become problematic. And that's why we call it the bad cholesterol. But here's one thing. So when you get a standard cholesterol test, which is called the lipid panel, here's what it shows you. It shows you the total cholesterol, HDL cholesterol, LDL cholesterol, and triglycerides. And so your doctor looks at this LDL number and says, oh, your LDL is too high, we need to lower it, you need statins. But the problem is that one LDL number, it's actually representing the amount of cholesterol that these LDL particles are carrying. And it doesn't really tell you anything about the particle itself. So some LDL particles are large, fluffy, and completely harmless. That's just like the normal LDL particle that just goes and carries the cholesterol to the tissue. But sometimes it becomes small, dense, and potentially dangerous under certain conditions. But when you get your standard blood test, it doesn't tell you the difference. So you clearly have two people with the same exact LDL cholesterol number, but one person has all the safe type and the other person has all the dangerous type. So they have the same number on paper, but completely different risk for cardiovascular disease. So, as we saw, cholesterol is not good or bad, it's just a molecule and it's essential for life. What we call cholesterol in blood tests is actually lipoproteins, which transport the cholesterol. HDL and LDL are different vehicles, but they're carrying the same cholesterol. And not all LDL is dangerous, but your standard blood test can tell you if your LDL is the safe kind or the dangerous kind. So how do we know which one do we have? How do we know if our LDL could actually become problematic? Well, fortunately, there is a better test. It's called advanced lipid testing or lipid electrophoresis. So this test doesn't just give you a total LDL number. It actually separates your LDL into its different subtypes and tells you exactly which one you have. And so what we discovered with this test is that LDL can be divided into 7 different subtypes. And these subtypes fall into 2 main patterns. Pattern A, which is subtype 1 and 2, and pattern B, which is subtypes 3, 4, 5, 6, and 7. So just remember that pattern A is the safe normal one, but pattern B is the dangerous modified one. So let's start with pattern A. These are what we call the large, fluffy LDL particles. They're bigger, they're buoyant, and most importantly, they're not oxidized. Remember that, they're not oxidized. So do you remember those proteins we talked about? The ones that are embedded in the outer membrane of the lipoprotein? Well, in pattern A LDL, those proteins are still intact. They still function properly. They're functioning normally. Now let's look at pattern B. These are what we call the small, dense LDL particles. They're smaller and heavier, but most critically, they're oxidized or glycated, okay? Their structure changed. So here, the protein on the outer membrane of these particles have been damaged. They've been oxidized or glycated. And when those proteins are damaged, that means that the same LDL particle now can't properly be cleared from your bloodstream by your liver. Your liver is supposed to recognize this protein that is on the membrane, but when it's damaged, your liver can't recognize it anymore, and so they end up sticking around longer in your bloodstream and they're much more prone to oxidation. So this is exactly the LDL particle that is associated with arteriosclerosis. This is the one that is associated with heart attack and strokes. Pattern B is dangerous, remember that. And if your test shows that you have predominantly pattern B, that's a red flag and it needs to be addressed. So I wanted to show you this data from a study of about 2,500 patients who had already had a heart attack. So they were followed for 5 years, and researchers look at their survival based on whether they have pattern A or pattern B LDL. So look at this line. This is the pattern B group. These people had small, dense, oxidized LDL. And after 5 years, nearly 90% of them have died. So after one heart attack, if you have pattern B LDL, your 5-year mortality rates is around 90%. Now look at this other line. This is the pattern A group, the people with large, fluffy, unoxidized LDL. After 5 years, more than double the number of people were still alive compared to the pattern B. So they had the same event, they had a heart attack. And in the follow-up period, which was 5 years, if you had pattern A LDL instead of B, you had more than twice the chance of survival. Now, it's important to point out that the difference between pattern A and B is actually very, very small. We're talking about 1 to 2 nanometers. So the size difference itself, it's not really the biggest issue. So what matters most is what happened to those proteins on the membrane. In pattern A, the proteins are intact and functional. But in pattern B, the proteins are now damaged, oxidized, and glycated. And that changes the conformation of the protein. The size is different, but it's not really what drives atherosclerosis. So, if you're concerned about heart disease, if you have a family history, if your doctor has told you your cholesterol is too high, you need to ask for an advanced lipid test. So this is what it looks like. You can see it breaks down your LDL into all the different subtypes. And it will literally tell you clearly if you have pattern A or pattern B. And if you have pattern A, even with high total LDL or cholesterol, you're pretty much safe and you're not likely to have any cardiovascular disease. But if you have pattern B, then you need to take some action and change your diet. Right, so I think that all of this raises an obvious question. What makes LDL shift from safe pattern A to dangerous pattern B? What's causing this damage to the protein? What's oxidizing these particles? Well, the answer is definitely going to surprise you. And it's going to completely change what you think about what causes heart disease. But we're going to talk about that in the next video. So if this video opened your eyes to something that your doctor has never told you, share it with someone who needs to hear this. And if you haven't already, please subscribe to my channel and hit the notification bell, because in the next video, we're going to talk about how sugar and not fat is what damages your cholesterol and makes it dangerous.